Hi, I'd like to thank you for joining us for our presentation today, where we will be discussing using NSG mice to study infectious diseases. To briefly outline what we'll be discussing, we'll be going through the process of how we establish human immunity in the immunodeficient mouse model, NSG mice. Next, I'll highlight a variety of examples of human-specific pathogens that have been modeled in humanized NSG mice, including HIV, dengue virus, EBV, a bacterial sepsis model, as well as malaria. I'll also show you a number of NSG-based platforms that can be used for infectious disease research. And finally, I'll end with telling you where you can go to find more information regarding all of these models to support your research. So today's presentation focuses on the NSG mouse and how it can be used to study infectious diseases. The NSG, or NSG-gamma mouse, was developed by Dr. Lenny Schultz's lab here at the Jackson Laboratory back in 2005. You can see the official strain name is located at the very top of the page, and our stock number for it is 005557. And so you can visit the strain data sheet and find that original publication, as well as a number of NSG-related web resources, and I'll show you where you can find that information at the end of our talk today. So the NSG mouse is a highly immunodeficient mouse strain, and there are three features that contribute to this immunodeficiency. The first is the NOD background. NOD, or NOD, is the non-obese diabetic inbred strain from which NSG mice were created, and inherent to this strain is a deficiency in innate immune system, including the absence of hemolytic complement, reduced dendritic and natural killer cell function, defective macrophages, as well as the presence of the SERP-alpha allele that allows for better human hematopoietic stem cell engraftment. And this will become important later when we discuss the ways that we make humanized mice. The second feature of NSG mice is the SCID mutation. SCID, or severe combined immunodeficiency, results in the loss of function of a gene involved in resolving double-stranded DNA breaks. And this is important during VDJ recombination during T and B cell development. So the presence of this mutation prevents mature T and B cell development, further adding to the immunodeficiency of this strain. The third and final feature of NSG mice that contributes to their immunodeficiency is the knockout of the IL-2 receptor gamma chain. This knockout eliminates signaling from 6 different interleukin cytokine pathways, and more importantly, it blocks natural killer cell development. So the IL-2 receptor gamma chain is a component of 6 different cytokine receptors, including IL-2, 4, 7, 9, 15, 20, and 21, as it's depicted by the blue subunit in this cartoon, with IL-15 signaling being involved in natural killer cell development. Completely knocking out the function of IL-2 receptor gamma results in no natural killer cell function, and this is actually one of the biggest impediments to hematopoietic stem cell engraftment in mice. So before I get into the details of how we build a humanized NSG mouse, I'd like to briefly go over the components of the immune system and how they all work together. There are two main branches of immunity, innate and adaptive. Innate immunity is the first line of defense against foreign invaders, and it does this through recognition of conserved molecular patterns commonly found on pathogens. Cells of the innate immune system include complement, which aids in the destruction of extracellular bacteria, macrophages, which engulf dead cells and pathogens, granulocytes, including neutrophils that release granules to directly kill microbes, and natural killer cells, which directly kill infected cells. The adaptive branch consists of B lymphocytes, and these make antibodies that aid in pathogen detection and engulfment, T cells that kill infected cells and help B cells make antibodies, and finally dendritic cells that present foreign antigens to other immune cells. The common link between these two branches are cytokines. All immune cells release cytokines to communicate with each other and respond appropriately to infection. And so as you can see, the NSG mouse is highly immunodeficient in a number of these different immune cell populations, including complement, natural killer cells, B cells, and T cells, with reduced macrophage and dendritic cell function. So some important features of the strain are that it has a longer lifespan than NOD/SCID mice. NSG mice can live as long as 16 months as compared to their NOD/SCID counterparts that typically develop thymic lymphomas and die at about 9 months. The SCID mutation also has some side effects. So in addition to inhibiting VDJ recombination, the SCID mutation globally affects DNA repair, so that any treatment that damages DNA, including radiation and genotoxic drugs, will require lower doses due to increased sensitivity. And finally, NSG mice can be used as a platform for creating more refined models, including transgenics and knockouts, and I'll show you some examples of this at the end of our presentation today. So as you can imagine, with the NSG mouse being so immunodeficient, it can be used for a variety of research areas, including cancer studies, human hematopoiesis and immunity, infectious diseases, as well as for the development of humanized mice. So what are humanized mice? In this setting, we refer to humanized mice as being mice that express a human immune system, and there are several ways to doing this, but here at the Jackson Laboratory we use different methods. The first is the hematopoietic stem cell engraftment, or the human CD34 positive cell engraftment model, in which we take human hematopoietic stem cells isolated from fetal liver, peripheral blood, or cord blood, and inject them via tail vein into recipient NSG mice. And then 12 weeks later, the human immune system has reconstituted in this mouse. And so some of the benefits for using this model is that it's capable of supporting multilineage hematopoiesis, and that's because the hematopoietic stem cells retain their stemness and are able to differentiate into all cells of the immune system. Also, these mice can be used for long-term studies, and that's due to the fact that a relatively naive human immune system that's implanted into this mouse tolerates the mouse host and doesn't recognize it as foreign. Some considerations for using this approach are that although we see robust reconstitution of T cells, their functionality is different than it would be in a human due to the fact that they are educated in a mouse thymus in the context of MHC and not HLA. HLA, which is human leukocyte antigen, is the human counterpart for mouse MHC, or major histocompatibility complex. Another limitation is that IgG responses and production are poor, and this is due to the lack of germinal center formation, appropriate germinal cell formation, as well as poor antibody class switching bonds. The last model that we use to create humanized mice is the PBMC engraftment model, in which peripheral mononuclear cells are injected intravenously or intraperitoneally into NSG mice. And so the benefits to using this model is that it is easy to establish, and it doesn't require any surgery or any additional time for reconstitution. Also, mature T cell populations, including effector and memory T cell subsets, are well established, and studies can be started soon after injection. Some considerations for using this model are that multilineage engraftment is absent, and that's due to the fact that human hematopoietic stem cells is not an enriched population in the peripheral blood. And so you won't see these populations well represented in this model. Another one is the lack of a primary antibody response, as well as the lack of HLA-restricted antigen-presenting cells. And this is due to the fact that this population is also not represented in very high numbers in the peripheral blood. And finally, after about a month or so, these mice will develop GVHD, or graft versus host disease, and that's because the human T cells in the peripheral blood will recognize mouse antigens as foreign and mount a response. So an improvement on the CD34 NSG model is the BLT model, or bone marrow, liver, and thymus model, in which human CD34 cells are injected into an NSG mouse, and then thymus and liver fragments from the same donor are also implanted into the subrenal capsule of that recipient NSG mouse. And so some of the benefits for using BLT model are similar to the CD34 model, that it does support multilineage hematopoiesis and can be used for long-term studies, but also because there are human fragments of thymus tissue implanted into these mice, that developing T cells in these mice are HLA restricted, thereby enhancing T cell activation and adaptive immune responses. They also have more robust mucosal immune engraftment. So some considerations for this model, using this model, is that it is a surgical model, and so it will require a little more time and expertise. And similar to the CD34 model, it does have a poor IgG response. So now that I've shown you the different approaches that we use to establish a human immune system in NSG mice, I just would like to highlight some key features of these humanized models. So these mice have the most efficient engraftment of human hematopoietic stem cells compared to other models. And that's due to the fact that these hematopoietic stem cells can home to the bone marrow and differentiate into all major immune cell types, including cells of the myeloid lineage, and that includes erythrocytes, monocytes, macrophages, dendritic cells, and granulocytes, as well as cells of the lymphoid lineage, including B cells, T cells, and natural killer cells. Also, the NSG mice, humanized NSG mice, do display some mucosal and adaptive immune function, and we'll see some examples of this later on in our discussion. So there have been comparative studies comparing human immune cell engraftment in NSG mice versus other immunodeficient mouse models, and each of them have shown that NSG mice is a superior host for human immune cell engraftment when compared to other strains that express the scid mutation alone, and that includes the original NOD-scid mice, as well as B6 scids, or the CB17 scid, which is the strain that the scid mutation originally spontaneously developed. NSG mice also have better engraftment than scid beige mice, as well as strains of different backgrounds that have the same or similar mutations, including the scid mutation or the RAG1 or 2 mutations that essentially knock out T and B cell development, or the gamma mutation eliminating natural killer cell function. So here's an example of one of those comparative studies in which NSG mice and NOD-scid mice were engrafted with CD34 positive hematopoietic stem cells, human hematopoietic stem cells, and then 10 weeks later their spleens were taken out and stained for human CD45, which is a pan-leukocyte marker. And we can see clearly here that at low MAG and high MAG, that NSG mice have much higher degrees of brown staining, indicating that they have much higher human immune cell engraftment compared to the NOD-scid mice. So this demonstrates that NSG mice is much better allowing for engraftment of human immune cells than its NOD-scid strain that it was developed from. In another study, NSG mice were compared to BRGs, and BRGs are similar to NSGs except that they are on the BALB-C inbred strain background. And you can see clearly here that NSG mice have much higher efficiency of human immune cell engraftment in the peripheral blood as compared to BRGs, indicating that the strain background plays a very important role in immune cell engraftment potential. So here at the Jackson Laboratory, when we engraft human CD34 cells, we take 3-week-old mice, NSG mice, and sublethally irradiate them and inject them via tail vein with CD34 positive hematopoietic stem cells from human. And then after about 12 weeks, we'll see B cells appear, and 15 weeks human T cells will be present. And so at this time, after reconstitution has fully completed, you can then go and measure immune cell engraftment, usually by flow cytometry. And so that's exactly what we did here. We measured engraftment in the peripheral blood, and we can see that human CD34 hematopoietic stem cell engrafted NSG mice had very high levels of human immune cell populations in the peripheral blood. Now I will note, you will see that there are some residual mouse immune cell populations in the peripheral blood, and that's mainly neutrophils and monocytes. But if you look down on the bottom right-hand corner, you will see that there are mature human T cell populations as well as B cells in the peripheral blood, and that's a great distinct advantage to using the NSG mice compared to NOD-scids, because this has particularly been a difficulty in seeing these populations in NOD-scid mice that were engrafted with CD34 cells. Also, when looking in the bone marrow, we see multilineage differentiation in the bone marrow. Not only do we see the rare hematopoietic stem cell population, but we also see human antigen presenting cell populations, as well as IgM producing B cells and human erythroid cell populations in the bone marrow. I will note that in the peripheral blood, we don't see human erythroid cell populations to very high numbers, but towards the end of our discussion today, I'll actually show you one way of getting around this limitation so that you can study pathogens that infect human red blood cells. So if you're interested in using any of these models, we have them readily available, and you can rely on our expertise for developing these models. So we routinely engraft cohorts of CD34 models and CD34 NSG mice, and we validate them to ensure that they have at least 25% of human immune cells in the peripheral blood. And this is the lowest that we'll accept, but typically we see between 60 to 80% human immune cell engraftment in the peripheral blood. These models are HLA typed for cancer research, and so you can take advantage of that. And we also do have PBMC models available once you place an order. So now that I've gone through the process of how we develop human immunity in NSG mice, let's go through some examples of infectious diseases that have been modeled in humanized NSGs. Let's start with HIV. HIV is a human-specific lentivirus that causes acquired immunodeficiency syndrome, in which infected individuals become severely immunocompromised and susceptible to opportunistic infections and cancer. Primate models typically have been used to study HIV, but as of late, humanized NSG mice have become the new go-to model for studying HIV. So in this particular example, the authors used NSG BLT models and compared them with non-scid BLT models for human immune cell engraftment, and you can see that in almost every immune cell population that they looked at, the NSG BLT models had much higher levels of engraftment, and that includes total white blood cells, so CD45, as well as CD4 positive T cells, CD8 positive T cells, and monocytes. Now, because HIV infects CD4 positive T cells, and because they see higher levels of engraftment in the NSG BLT model of this population, they decided to see if this correlates with higher infectivity. So they intravaginally inoculated NSG BLT mice and non-scid BLT mice with HIV and then measured plasma HIV viral loads. And you can see that in the non-scid BLT mice had much lower levels of HIV RNA, barely beyond detection, as compared to NSG BLT mice that had much higher levels of HIV viral RNA, indicating that NSG BLT mice are capable of supporting HIV infection. So they also did some staining for CD3 total T cells and CD4 positive T cells in the vaginal mucosa of non-scid BLTs and NSG BLT mice, and they see much higher levels of staining of total T cell populations in the NSG BLT mice as compared to non-skids, and they saw the same for the CD4 positive T cell populations, indicating that not only can you use this model for studying HIV infection, but also for studying human immune cell responses in response to HIV infection. So one application for using humanized NSG mice is for designing and testing new treatments. In this example, NSG BLT mice were intravaginally infected with HIV and then treated with antiretroviral therapy or ART therapy that's typically given to human patients. And so you can see here that when they measured HIV viral RNA in the plasma, that when ART therapy was not begun, you see high levels of HIV RNA in the plasma, and then when ART therapy was administered, you see a dramatic reduction in viral loads, and you can see that in panels C and D. And when ART therapy was stopped, that these viral loads rebound to pre-treatment levels. They also collected viral RNA from peripheral blood, lymph nodes, and other tissues, and they saw low levels of HIV RNA, indicating that these cells were latently infected with HIV, and that mirrors what's seen in humans. So together, this data nicely shows that not only do NSG BLTs recapitulate human HIV infection, but also that it's a good model for testing the efficacy of new treatments. So another utility of NSG mice in HIV research is to use it to test different gene therapies. So in this particular example, they used a common readout for HIV infection, which is CD4 positive T cell loss. Because CD4 positive T cells are infected by HIV, it's common to see a reduction in this population due to HIV infection. So they use this as a readout for testing the efficacy of a gene therapy in which human hematopoietic stem cells were engineered to express a transgenic T cell receptor that recognizes the SL9 gag protein of HIV, so that CD8 positive T cells can directly kill HIV-infected cells. And so you can see here that in the SL9 T cell receptors, that transgenic T cell receptor mice, they also were BLT models. You can see much higher levels of CD4 positive T cell populations as compared to the NSG BLT mice that express the control T cell receptor. This also correlated with viral loads in the peripheral blood. You can see much lower levels of viral RNA in the transgenic T cell receptors that recognize HIV as compared to the mice that express the control T cell receptor. And they saw this in the spleen, bone marrow, and the thymus. So together this indicates that not only can you use this NSG BLT model to study HIV infection, but you can also use it to develop new gene therapies as a strategy for anti-HIV treatment. So NSG mice can also be used to develop and validate new prophylactic treatments. In this study, mice were administered an adeno-associated virus that expresses a broadly neutralizing antibody against HIV, and then they were injected with human peripheral blood mononuclear cells and then infected with HIV by intravenous infection. So before they did this, they tracked CD4 positive T cell populations in non-immunized mice, and you can see that HIV-infected mice show the characteristic drop of CD4 populations as compared to the mock-infected. And then when they infected these mice with HIV, you can see that even in the presence of increasing amount of HIV, that the mice expressing the neutralizing antibody against HIV were protected from the CD4 positive T cell loss, as compared to the luciferase control, where you do see the loss of these populations. So together, this data nicely demonstrates that these mice can be used for developing new prophylactic treatments. As we've seen so far, humanized NSG models can be used to recapitulate several aspects of HIV infection in humans. Some secondary complications of HIV infection also include the reduction of neuronal integrity. And so for these studies, the authors wanted to determine if this could be modeled in NSG mice. So NSG mice were engrafted with human CD34 cells at birth and then infected with HIV 22 weeks later. And then they measured CD4 positive T cell populations and neuronal integrity. And so you can see that CD4 positive T cell populations were dramatically reduced in HIV-infected mice as compared to control mice. And then when they looked at two readouts for neuronal integrity, MAP2 as well as neurofilament protein, you can see that in the HIV-infected mice, there's much lower degrees of staining for MAP2 and neurofilament proteins as compared to uninfected mice. So this demonstrates that not only do NSG mice have the ability to recapitulate primary complications of infection, including CD4 positive T cell loss, but they also can demonstrate secondary complications such as neuronal integrity. So now I'd like to switch gears a bit and talk about dengue virus infection modeling in humanized NSG mice. So dengue virus, which is the causative agent for dengue fever and the more serious dengue hemorrhagic fever, is a global health concern, and it's actually been difficult to study in non-human animal models because only humans typically demonstrate the clinical symptoms, including viremia, erythema or redness, as well as thrombocytopenia or reduction in platelet count, as well as fever. And so in these particular experiments, the authors wanted to show whether or not NSG mice can be used to recapitulate some of these clinical symptoms only seen in humans. So they infected CD34 NSG mice subcutaneously with 4 different dengue virus genotypes and looked at 4 different clinical parameters. And so when they looked at viremia in the blood, they see high levels of viremia for all of the genotypes that were administered, indicating that NSG mice are capable of supporting infection. They also looked at erythema or redness, and they see that much higher degrees of redness for the dengue virus infected mice as compared to the control. They also saw higher levels of fever or body temperature, and you can see this spike in body temperature post-infection as compared to the control. And when they measured thrombocytopenia, they saw that all of the infected mice showed reduced platelet counts. As we can see over time, platelet counts reduce during infection with the Southeast Asian strain. And when we look at panel B, you see that all of the genotypes that were used to infect NSG mice all had lower platelet counts as compared to the control, indicating that NSG mice can be used to study dengue virus infection and that it shows similar clinical symptoms that have only been seen in humans. So not only do NSG mice recapitulate similar clinical symptoms, but they also can be bitten by mosquito and infected that way. So in these studies, the authors compared viral titers and erythema from human CD34 engrafted NSG mice that were infected either subcutaneously with dengue virus or by mosquito bite. And so we can see here that mice that were infected with mosquito bite have a much more prolonged course of infection as compared to the subcutaneously injected. And then when they measured erythema, the mosquito-bitten mice had much higher levels of redness. So together, this data just demonstrate the utility of using these models in studying this disease with a more natural route of infection, and you can study how these factors play a role in disease progression. So for my last example of dengue virus infection, I'd like to show you how humanized NSG mice can be used to measure T-cell responses and antibody production in response to infection. As I mentioned previously in our presentation, you can enhance T-cell responses in these models by engrafting human thymus fragments to HLA restrict developing T-cells. Another way to HLA restrict is by expressing HLA transgenically, like the authors did in this study. NSG BLT mice and human CD34 engrafted NSG mice that expresses an HLA class 1 were infected subcutaneously with dengue virus CIRVAR2, and IgM production was measured as a readout for T-cell activation and response. We can see that both the NSG BLT mice as well as the HLA transgenic NSG mice had much higher levels of IgM production as compared to NSG mice that were not HLA restricted. So this indicates that HLA restricting these NSG mice can be used to develop more robust T-cell responses in response, and you can measure these responses during infection. So now I'd like to show you some examples of Epstein-Barr virus infection modeling in humanized NSGs. EBV is one of the most common viruses found in humans, and it causes infectious mononucleosis, Burkitt's lymphoma, and Hodgkin's lymphoma. EBV establishes a latent infection in B cells, and it has the ability to transform these cells to cause cancer. Currently, there's no vaccine, and it's been difficult to study in other models, so it's important that we develop a model to study this infection. In this particular publication, the authors infected CD34 engrafted NSG mice intraperitoneally with EBV isolated from human saliva, and then stained for EBV-encoded RNAs in various organs to measure EBV localization. So when they looked before weeks post-infection in the spleen, lymph node, and the liver, they detected EBV in each organ. And when they stained for human T cells in the spleen and kidney, they also found EBV to co-localize with these T cells. So they took a look at these T cell populations, and not only were they in close physical proximity of the virus, but they also were more activated in response to infection and expanded in response to infection. And you can see that EBV infected total T cells as well as CD8 positive T cells are expanded as compared to the control, and they're the activated memory T cell phenotype. And represented graphically, you can see much higher levels of T cells during EBV infection as compared to the control, and it's mostly due to its CD8 positive T cell expansion. So as you can recall, HLA restricting humanized NSG mice leads to enhanced T cell responses. So when the authors used humanized HLA-restricted transgenic mice to measure interferon-gamma secretion as a readout for T-cell response to EBV infection, they saw that during lytic or productive infection, only HLA-restricted T-cells produce interferon-gamma and not the MHC-restricted. So yet this is another example of how HLA restriction of T-cells can lead to greater and more robust T-cell responses in these models. So as I mentioned previously, EBV causes B-cell lymphomas, and the authors use tumor growth as a readout for T-cell function during EBV infection. So they depleted T-cells using a neutralizing antibody against T-cells, or they used an isotype control, and then measured tumor growth in the spleen, kidney, mesenteric lymph node, and the liver. And you can see here that in mice that were depleted for T-cells developed tumors in each of these different organs. They also looked at viral loads in the spleen, and they saw that administering these T-cell depleting antibodies resulted in higher EBV loads in the spleen, indicating that T-cells are part of the response and fighting EBV infection. So together, this data from this study just really elegantly demonstrate that humanized NSGs can be used to not only recapitulate human pathology associated with EBV, but you can also measure immune responses and study immune responses to EBV infection. And ultimately, you can use it as a preclinical model for studying EBV. So now that we've gone through some viral pathogens, let's go through an example of a bacterial sepsis model. Sepsis is a major health concern, and although it can be caused by systemic infection of viruses or fungi, bacteria are the most common contributors. So sepsis is commonly modeled in mice via a cecal ligation and a puncture technique, which releases any cecal-associated pathogens into the sterile intraperitoneal cavity, and this is typically dominated by bacteria. So doing this triggers a systemic inflammatory response and the release of cytokines known as a cytokine storm. So in this study, the authors compared BALB-Cs, which is an inbred strain, NSGs, and NSG BLT mice that were subjected to a cecal ligation and puncture, and then they measured human cytokine production in the peripheral blood. As we can see, for all of these inflammatory cytokines that they looked at, they are much higher in the humanized BLT mice as compared to the BALB-Cs or the non-scid mice. And also, when they inhibited a key inflammatory molecule, HMGB1, via siRNA, and then measured human cytokine production in the peripheral blood, they saw a dramatic reduction in all of these cytokine levels. And this was only seen in the human cytokines and not the mouse cytokines. When they also measured survival, administration of this siRNA against HMGB1 greatly improved survival. So together, all this indicates that you can use NSG BLT mice as a preclinical model for studying bacterial sepsis. So finally, I'd like to end the data portion of our discussion by looking at malaria disease modeling. So I mentioned at the beginning of our discussion that human erythrocyte populations in the peripheral blood are low after immune cell reconstitution with the CD34 model. But one way to get around this limitation is to daily inject human erythrocytes intraperitoneally until you reach a point of saturation. And then you can use these models to study pathogens that typically infect human red blood cells. And so that's exactly what the authors did here. NSG mice and NSB mice, NSBs are beta-2 microglobulin mice that reduces natural killer cell function. And so they engrafted these mice with human erythrocytes and then subsequently infected them with 2 strains of Plasmodium falciparum. When they measured erythrocyte populations between these 2 models, they see similar engraftment efficiencies. However, when they look at parasitemia, you see a dramatic reduction in parasitemia levels of NSB mice, and that's because they are more immunocompetent than NSG mice, and they're better able to clear infection. I will note that these NSG parasitemia levels are similar to thresholds that are commonly seen in humans. So this indicates nicely that NSG mice that are engrafted with human erythrocytes are capable of supporting malaria infection. So the authors in this study took it a step further and used these models to test standard of care against malaria treatments, as well as a novel anti-malarial compound. And so you can see here with increasing doses of these standard of care agents that we see reduction of parasitemia. Also, when they tested their new novel anti-malarial compound, you see much lower levels of parasitemia in response to higher doses of their anti-malaria compound. So these studies validate the use of humanized NSG mice as a preclinical model for testing new anti-malarial therapies. Now that I've highlighted the benefits of using NSG mice as a model for several human-specific pathogens, I'd like to tell you about some newly developed strains that are related to the NSG mouse and how they are improvements on that model. So the first of these is the NRG, or the NOD-RAG-gamma mouse. This strain is an alternative to the NSG mouse that is radiation resistant because it carries the RAG mutation in place of the SCID mutation that results in a lack of T and B cell development. NRGs do have comparable hematopoietic stem cell engraftment levels to NSG mice, but they are radiation resistant. So if you're doing any experiments that you need to irradiate mice, or if you're using genotoxic drugs, you might want to consider using this model in place of NSG mice. Some other strains that are also based off of the NSG mouse are the NSG beta-2 microglobulin mice, which lack MHC class 1 function and allows for better PBMC engraftment. We also do have some HLA class 1 transgenics indicated here, and actually the blue numbers are our stock numbers. Type in these links to go to the strain data sheets for each of these strains. We also have HLA class 2 transgenics. We have the DR4 as well as DR1s and DRAG mice, and all the stock numbers are indicated here. And finally, we have these other transgenic mice that express KIT ligand and do not require preconditioning irradiation, as well as the second strain that expresses human IL-3, CSF2, and KIT ligand. And these mice can be used to study human myeloid cell development. And finally, if you're doing any transplantation studies, you can use this third one that expresses GFP in all cells, and you can use GFP to track your transplanted cells. So finally, I'd like to show you where you can find more information regarding all of these models. So the NSG mice strain data sheet is located here. It's stock number 005557, and on that strain data sheet you can find a wealth of information including genotyping protocols, references including the primary reference from Dr. Lenny Schultz's lab, as well as this resources for the NSG mouse model. So if you click on that link, it'll bring you to this page here where you can find additional links including the breakthrough research for NSG, our NSG mouse model portfolio, as well as FAQs. And as I mentioned, because NSG mice are so immunodeficient, they can be used in a number of different disease areas, including immunology, infectious disease, as well as cancer. And so you can find a number of references relating to these disease areas with our annotated NSG references list that's updated every month with new references. So as I mentioned, if you're interested in using these humanized NSG mice, you can order them from us where you can rely on our expertise as well as save yourself some time, because we routinely engraft the CD34 model about every 6 weeks or so, and after 12 weeks we validate these mice to ensure that they have at least 25% human immune cell engraftment, but typically we see between 60 to 80% human immune cell engraftment in the peripheral blood. PBMC models available. If you'd like to make your own humanized models, there are several published protocols that can help get you started, and some of these are listed here. With that, I'd like to thank you for joining us for our presentation today, and just wanted to remind you that not only do we sell mice, but we have a number of different mice-related services, and so you can take advantage of our expertise in performing some of these services, including highly customized and complex breeding projects, as well as compound efficiency studies. So if any of these things are of interest to you, please feel free to contact us or visit our website to find out more information. So I'd like to thank you again for joining us for our presentation today, and if you have any questions, you can always contact your regional representative by calling them or emailing them, or you can contact me or my colleagues in technical information for any mouse-related questions you may have, visiting our tech support portion of our webpage. Thank you so much.